just step aside for a minute and say that there were some people who did not believe these results. So there were people who argued that when these experiments were done, whether it was Gurdon's or Dolly or more so Gurdon, that when they isolated the cells, that there must have been some stem cells hidden in there. So stem cells that were not differentiated. And that's how you managed to make these new organisms. So that, again, the argument that an adult cell is too far along and that you have to get something that is at a more pluripotent or multipotent state. And so Rudy Janisch, from Harvard, MIT, sorry, designed an experiment with one of his postdocs to test that idea, or to find a way to really establish that an adult cell could be used, the nucleus of an adult cell, for cloning. And so they took advantage of the fact that lymphocytes, which are immune system cells, when lymphocytes are formed, so they start out with the same genome as all the cells in the body, but their DNA gets rearranged in certain regions. And that rearrangement can serve as a marker for those cells. So those cells have now different DNA than the rest of the body. Janisch and Hochedlinger, it's a hard name to say, they took the nucleus from one of these lymphocytes, and this is in mice, and they used it to clone a mouse. And then when they made adult mice in this experiment, they then looked at their DNA and they saw that they carried exactly those rearrangements that were present in those lymphocytes to begin with, effectively proving that the DNA from an adult, terminally differentiated, so at the end of its developmental stage,